in Pasadena, which is near Los Angeles. I'm going to quickly share in the chat my contact information, if I can figure out how to do that, my email. So I'm going to do a couple of different things in the hour that I've been given. I'm a stable isotope geochemist. That means that I study all kinds of natural processes, all kinds of natural phenomena, using the distribution of the naturally occurring common stable isotopes, meaning the isotopes that don't undergo radioactive decay. I'm not going to spend any time this hour talking about very technical subjects like measurement technologies or the strange nomenclatures that people in this specialty use to report data. We're not really going to get too deeply into quantum mechanics. What I want to get across is concepts. This is a discipline that will seem unfamiliar to some of you. A few of you will have encountered this before. Maybe you've run across stable isotope data in papers. Maybe you've had a class at university. But for the most part, it's something that seems far away from most of our applied interests, if we're environmental scientists or marine scientists or climate scientists. But when you scratch the surface, you discover that data of this type are everywhere. They're the foundation for lots of different arguments that we construct in studies of ecology, marine circulation, ocean circulation, climate history, recent climate history, deep time climate history, atmospheric chemistry. All of these disciplines make use of stable isotope data. And so I've created a lecture that gives you a little taste of different sorts of concepts that come into play in the use of the stable isotopes, stretching from very old tools that have been around for nearly a century, all the way up to things that were just invented within the last 6 months, literally. So just sit back, enjoy it. I'll hopefully finish a little bit early, so we'll have some time for discussion and questions. If you have further follow-up questions or things you'd like to talk about, feel free to email me. I'm like old and don't understand Slack, but I'll see if I can figure out how to join the Slack later in the week. And then finally, I'd like to point out that Caltech, California Institute of Technology, my home institution, there are a variety of ways for students who are not already part of our campus to get involved in some way with things that we're doing. If you're an undergraduate or have nearly finished your secondary school training, we have summer undergraduate research fellowships that'll bring people to campus for 10 weeks to do research projects in a professor's lab. The undergraduate program, if you're interested in applying, you can talk to me for guidance. Graduate program in the Earth Environmental Sciences, I can also be a point of contact. Postdoctoral fellowships, those of you who have a PhD already, I can give you some guidance how to apply for those sorts of positions. And then if you want to visit, just as a visiting scholar of any level of seniority, you want to come and have a research experience, you're not taking classes or whatever, you're just here doing science, I can also help set you up. So send me an email if any of those things interest you. So without any more preamble, let's kick this thing off. So I think all of us, no matter what your background is, all of us have some sort of experience of chemistry, of natural chemistry. There's something like 52 million compounds known to science. Almost all of these are organic molecules. That gives you one immediate insight. All of the diversity in the chemical world is organic, and only a small amount of it is inorganic or minerals in the geological record. And there are many different areas of science that are concerned with describing the diversity, the structure, the properties, the chemical reactivity of all of these different compounds. You encountered this through chemistry classes early in your education. Maybe you had a physics class that talked about the dynamics, the kinematics, the motions of atoms within molecules. Engineers and environmental scientists describe them. Most areas of science think of the naturally occurring molecules as sort of like little machines. They're things that do something or that have a certain structure or property. And that's sort of the end of your question about them. Natural scientists, earth scientists, we have a wide variety of other questions that we bring to molecules. We don't want to know just what it is structurally or how it behaves mechanically. We want to ask it questions you might ask of a book that you pick up off a shelf. We want to read molecules as a record of some kind of natural history that is otherwise hidden from us, obscured from us. We might want to know what a molecule was made from and what kind of chemical reactions made it. We might want to place that molecule in a chronology, in a natural history. We might want to date it or know when it was made. We might want to know geographically where it has been on the Earth or to what depth in the ocean was it synthesized. We might want to quantify the conditions that it experienced, like the temperatures at which it was made or the temperatures at which it was stored. We might want to describe processes that changed it after its formation, like an atmospheric process that partially destroyed the compound that we're interested in. And then finally, we might have very specific forensic questions. We might want to identify whether a molecule does or doesn't belong to a class of organisms or even an individual being, individual person. So these sorts of questions, the reading of molecules as a record of their history and of their environment, it's a very strange concept, but there's something that is familiar. It's part of our daily life now that I think gives us some insight into how it might be approached. So, and that's genetics. So all of us know, if you read a newspaper or you've had any kind of science training or you've been to a doctor, you know that the analysis of the human genome is a really key component of describing human health and all kinds of other issues. So why does this work? The reason why genomics is a record of such detailed and nuanced information is its tremendous structural complexity and diversity. And this, the structural complexity is sort of part of the way that it functions. The diversity emerges through random mutation and inheritance. And then this record becomes readable to us through gene sequencing. And it's really the combination of these things: great structural complexity, diversity that arises through mutation and inheritance, and then an analytical technology that lets us read that structural complexity. That's what underlies the use of genomics in forensics, like criminal forensics, disease diagnosis, modification of the genome for treatment, genetic engineering for various purposes. All of it rests upon this observation of complexity and diversity. So when we go to the other 52 million molecules that are known to science, they seem kind of bland in comparison. They don't really seem like something that we could approach in this way, where the molecule is a record of its history. Here's a suite of organic molecules. All of them are important. Just like you can't live without your DNA, you can't live without your sugars and fatty acids. I can't live without my caffeine. A car doesn't run without its isooctane. So all of these are really important things, but they're structurally simple. They might seem more complicated than like a single atom or a dimer, two atoms stuck to each other, but compared to DNA, they're really, really structurally simple. And more importantly, the concept of mutation and inheritance really isn't part of the chemistry of these compounds. Every single, say, glucose molecule in your bloodstream, they seem basically the same. They're the same structurally. They might have little isomeric transformations of their shape, but they're all the isooctane molecules are basically the same. All the caffeine molecules are basically the same. They seem anonymous. How could I possibly read a difference in history between different samples of these molecules if they are identical? So the isotopes are what allow us to read, to individuate, to separate the histories of different samples of molecules. So let's begin by reminding ourselves about the major components of atomic structure. So this is a cartoon of the carbon atom, of carbon-12. And the dense nucleus, where all the mass sits, is composed of 6 protons, so 6 positive charges, 6 neutrons. They contribute nothing to charge, but they do add to mass of the nucleus. And then the orbital electrons that really control chemistry. So one of the key concepts in our understanding of chemistry and of the behavior of atoms is that the number of protons dictates the charge at the center of an atom. That controls the stable electronic structure of that atom, and it's electronic structure that controls chemistry, controls atomic behavior. And this concept is how we arrive at the structure of the periodic table of elements. The periodic table of elements is basically organized according to electronic structure and therefore chemical behavior. That electronic structure reflects the number of protons in the nucleus, and that's really the only information that you really care about for normal chemistry. The neutrons, which do not contribute to charge, don't really contribute to the electronic structure. They're sort of irrelevant. Isotope is a word that was a made-up word. It was made up around 1920, means same place in the periodic table of elements. And what it refers to is the phenomenon of multiple atomic species that share the same number of protons, and therefore they share the same number of electrons, and therefore they share the same chemistry, and they end up at the same place in the periodic table, yet they have different numbers of neutrons. The simplest example you can think of are the isotopes of hydrogen. So the hydrogen atom normally is just a proton with an electron orbiting it. Deuterium, the natural stable isotope of hydrogen, has a single extra neutron in its nucleus, making it twice as heavy as a normal hydrogen atom. Stable means it doesn't undergo radioactive decay. Tritium, an extremely rare isotope of hydrogen, has 2 neutrons within its nucleus. That makes the nucleus unstable, and it spontaneously undergoes radioactive decay with a half-life of a little more than a decade. So 3 different isotopes of hydrogen, all of them behave chemically like hydrogen, and yet they're different. Different masses, one of them is radioactive, and these differences in the nuclear properties lead to emergent differences in their abundances in natural materials. So there's a concept I'm going to introduce now, but we'll really only come back and try to take advantage of it at the end of this hour. And that is the concept of what we call isotopologues. An isotope is a version of an atom that differs in numbers of neutrons from its more abundant, more common relative. An isotopologue is a version of a molecule that has a different mixture of isotopes within its structure. So consider table sugar, sucrose. So sucrose has 12 carbon atoms and 12 oxygen atoms and 24 hydrogen atoms. And now let's ask ourselves, what are all the different ways that the rare isotopes, stable isotopes of carbon, of oxygen, and of hydrogen could be inside this molecule? I could have a single rare carbon-13. It's about 1% of all carbon atoms. So there's like a 12% chance I'll have one carbon-13 in this molecule. Or I might have a 24 in 7,000 chance of a single deuterium atom somewhere in this molecule. Or I might have a 12 in 500 chance of a single atom of oxygen 18, a rare isotope of oxygen, it's somewhere in this molecule. There then are, but then we have to consider the different atomic positions in this molecule are not symmetrically identical. And if I put a rare isotope in one position, it might change the molecule in a way that's different from placing it in another position. We call these position-specific differences or position-specific isotope effects. The rare isotope makes a choice which of the atomic positions it will occupy. Another thing we have to consider is that there's a small but finite chance that I'll have more than one rare isotope in the same molecule. It's a strange sort of idea. If the isotopes are rare, a part per, a percent, a tenth of a percent of this order, then what are the odds that I would have two of them in the same molecule, or three of them, or four of them, or five of them? Well, it would be a vanishingly small chance. But of course, there's lots of molecules. A mole, 10 to the 23rd, that's a lot of molecules. So even things that are very rare, they occur. And the double rare isotope substitutions are part of that. I might have two rare isotopes that are next to each other, stuck to each other, and therefore closely interacting with each other, or they might be in the same molecule but in different positions. When you consider all of these possibilities, and then you sit down and calculate what are all the different versions of the table sugar molecule. There's 3 times 10 to the 15 of them. That's way more than the number of genes in the human genome. It's more than the number of stars in our galaxy. It's a huge, huge number. And a key concept is that the physical properties, the chemical properties, the probability of being made for all of these isotopic forms, all these isotopologues, all of them are unique. And so every one of them is, in a sense, an independent vote, an independent record of the history of that molecule. What this tells us is that the distribution of isotopes in molecules is a record of history that's just as rich, just as nuanced, just as complicated as genomics. We just have to figure out how to read it and how to interpret it. And that's my job, basically. That's what I do for a living for all sorts of different natural science problems. So let's now pull back from that complexity and just look at the way that rare isotopes have typically been used to study the origins of materials, where we're not trying to tell the differences between all these structural isotopologues. We're just trying to measure the abundance of the rare isotopes in a sample of some kind. And let's see how far this gets us. So here's an example from recent history where radioactive decay of isotopes is the key piece of information that we need. So the Chernobyl reactor in the Ukraine underwent a runaway reaction, a meltdown in 1986. And in the weeks after this occurred, there was very little direct, clear information coming out about exactly what had occurred. But samples of dust that were released from this meltdown were collected elsewhere outside of the Ukraine. And they were analyzed for their ratios of rare isotopes of iodine. Both of these are radioactive isotopes, but one of them, the iodine-133, it undergoes radioactive decay faster than iodine-131. And so as you sit and collect the dust and observe over time from one day to the next, you see the ratio of these isotopes dropping as the fast decaying one diminishes faster than the slow decaying one. By projecting back along the trend of this decay, and then looking at the intersection of that with the known production ratio, given the kind of reactor this is, you can place a date to within hours of when the disaster occurred. And this is the first way that we learned when the Chernobyl meltdown occurred. Here's another example where radioactive decay is part of the problem, but it isn't really directly how we're doing dating. So this is a plot that shows on the vertical axis the abundance of carbon-14 in atmospheric CO2, which is quickly inherited by organic matter in the Earth's biosphere through photosynthetic carbon fixation, and then animals eat all the different plants and things. So within a year or two, the carbon-14 content of the atmosphere gets imprinted on the biosphere. So there's a background level of carbon-14 that's naturally generated in the atmosphere through high energy particle collisions with nitrogen. And it's just sort of there, okay? And that background amount of C14 is the basis of normal carbon-14 dating that's used in ecology and lots of different other fields. But during the Cold War, so in the 1960s, there was a tremendous spike, basically a doubling in the carbon-14 content of CO2 in the atmosphere due to atmospheric nuclear weapons testing. And it rose exponentially. And then the atmospheric testing of nuclear weapons was banned, like in 1960 something. And there was a little bit after that, but basically that source shut off. The half-life of carbon-14 is over 5,000 years. So you would think that this elevated concentration would take a very long time to decay, but it didn't. And the reason why it decayed much faster than you would expect from radioactive decay alone is that the biosphere took up that carbon-14, distributed it through organic matter, dissolved it in the ocean as dissolved in organic carbon, and diluted it by the much larger amounts of carbon that were in the biosphere and dissolved in the ocean. And so it effectively rinsed that atmospheric spike out of the atmosphere in decades instead of over thousands of years. This then creates what's called the bomb spike. And the bomb spike can be used to date small differences in time that are relatively recent. So for example, the carbon atoms in my brain, I'm older than pretty much all of you, it are richer in carbon-14 by about 70% than most of yours, because those tissues were laid down when I was born and really have not been swapped out. Your cerebral cortexes have maybe 50% less carbon-14. Our bones swap out carbon atoms in the collagen very quickly because we have to repair damage to the bones. Our intestines, our guts trade out carbon atoms very, very quickly on weeks to months because they're a tissue that's very active, gets damaged quickly, regenerates itself quickly. So you can date different parts of your body and when they were last fixed from the external biosphere using carbon-14 content through the bomb spike. So these are two examples of the use of radioactive isotopes in order to learn things about the histories and origins of materials. But this is only a small fraction of all the information that you can get. The information that my research group focuses on, and I would say really it's a broader range of applications in the natural sciences, look at the stable isotopes, the isotopes that do not undergo radioactive decay. And for the most part, they're not produced by radioactive decay of other elements. They're just there. They've been there from the origin of the Earth, and they get redistributed through natural materials, through chemical and physical processes. We're not going to spend the rest of this lecture learning about those sorts of processes, how they work, and what sorts of tools emerge from them. As a sort of warning, they're complicated. Okay, you really cannot deeply understand the separation of isotopes through chemical processes without really probing the depths of quantum mechanics. So we're going to graze across the surface of this, focusing on concepts rather than mathematics and sort of deep levels of theory. So here's one very simple, pretty easy to understand way in which isotopic identity changes a physical behavior of a material and therefore leads to a separation of isotopes. So consider a case where I have two versions of a molecule, one that has a heavy isotope with extra neutrons, like carbon-13, and one that has a light isotope that it lacks the extra neutron, like the common carbon-12. And then I expose some carbon-bearing molecule like CO2 to a small pore, say the pores in the leaves of plants that CO2 has to enter through before it can be photosynthetically fixed. Well, the velocity of these molecules is proportional to their kinetic energy, just through Newton's laws. The kinetic energy is proportional only to temperature. So at a given temperature, the light molecule is moving faster because it has a lower mass than the heavy molecule. And so it more easily penetrates through pores. This is an important part of the photosynthetic isotopic fractionation. Here's another case. It's less common geologically, but it's an important tool for understanding the history of climate. Imagine that I have a packed column of some sedimentary material, It could be glacial snow or fern, which is like densely packed snow. It could be a sand dune. Anything that is a densely packed aggregate of materials that have little fine pore spaces that cannot undergo convection. In cases like this, I might have a column of gas that's composed of heavy isotopes and light isotopes together. If I allow them to settle with no wind blowing through them, no convection overturning them, nothing stirring them, they'll spontaneously settle so that the heavy isotopes are preferentially distributed at the bottom and the light isotopes at the top. And we can use this signature to figure out how thick columns of gas were, because it takes time for the sorting to occur. We can sort of date the gases that are in densely packed columns of sediment, and there are other tools that emerge from this as well. So these first two examples that I gave you are simple physical fractionations of isotopes, separations of heavy from light isotopes that follow chemical principles that are really familiar to everyone who's had like a high school physics class. All of us know Newton's equations of motion. All of us understand that gravity pulls heavy things down and light things up. But now we're going to get into the isotope effects that are really driven by chemical reactivity and chemical stability. And these are isotope effects that are much more common. They're basically ubiquitous. They're found all through nature, but they are harder to understand. And a beginning of an understanding of them is to recognize that chemical bonds behave sort of like springs. Okay, they're effectively, you can think of molecules or minerals as being point masses of atoms that are joined to each other through springs. And the springs are the electrons that are forming chemical bonds. So here's an example of a molecule, a really simple molecule, hydrogen chloride. In one version, the H is a normal hydrogen atom. In the second version, the H is the rare deuterium isotope, the heavy isotope of hydrogen. When I look at the frequency of the motion, the spring is the same. Okay, the spring that connects the atoms to each other is the electronic structure of the bond. In one case, the spring is acting on something light. On the second case, it's acting on something heavy. It's like if I had a slinky spring and I put a tennis ball on the end, it's going to bounce quickly. If I put a bowling ball on the end, it's going to bounce very slowly. So we know that then the heavy isotopes have lower frequencies of motion of their chemical bonds. We know through Planck's postulate that the energy contained in a chemical bond is proportional to the frequency of the movement of the atoms. Energy is proportional to frequency. This is a founding concept of quantum mechanics and really one of the ideas that launched the physics of the 20th century. The combination of these two ideas tell us that when I put a higher mass rare isotope into a molecule, I lower the frequency of the molecule's motions, and therefore I lower its energy content. I make the molecule more stable. And so a key concept for all of the chemistry of isotopes is that every molecule you've ever seen, every mineral you've ever seen, has a thermodynamic desire to take in heavy isotopes. Every single molecule will become more stable, harder to break down, more persistent in the environment, stable to some higher temperature if you can put heavy isotopes into it. All the molecules are hungry for heavy isotopes. Here's an experimental observation that shows you the effect that I've just talked about. This is the visible light spectrum, and then around it, the infrared longer wavelength spectrum and the ultraviolet short wavelength spectrum of light. So if I look closely in the infrared spectrum, I'll see absorption bands in when I pass light through simple gaseous molecules like carbon dioxide. These absorption bands correspond to the wavelengths of light that match the frequencies of molecular vibrations. When incoming light matches a frequency of molecular motion, the two couple with each other, and the energy of the light can be dumped into the energy of motion in the molecule. It excites the molecule, and the light gets absorbed. When I do this on a version of the CO2 molecule that has a heavy rare isotope in it, I see all of these absorption frequencies shift to lower frequency, deeper into the infrared. And this is a simple experimental observation that tells you that this change in the dynamics of molecules occurs when you do heavy isotope substitution. Here's another crazy little factoid. It's hard to know exactly what to make of it, but it's one of the funner things I've ever encountered in studying stable isotopes, and I've done this a long time. So, and that is that isotopic substitution changes the way chemicals smell. Maybe not to us, I don't know if we're sensitive enough to notice it, but it works for flies anyway. It changes how things smell to flies and maybe other animals. So all of us are taught, at least sort of in a casual way, that the way our sense of smell works is like a key in a lock, that a molecule you can smell somehow fits chemically into a receptor that's in your nose. And when it fits into it, it excites it and you have the sense of smell. But then there was an experiment done that showed smell is not just a key in a lock. The motions of the molecule, the vibrations of the molecule also somehow contribute to smell. And the way this experiment was done was to take a molecule that flies normally love. It's called acetophenone, and it's something, for whatever reason, they think it smells delicious. And then the researchers took all the hydrogen atoms off and replaced them with deuterium, with the heavy isotope of hydrogen. The shape of the molecule is totally unchanged. That depends on electronic structure. But the motions of the molecule are slowed. And the slowing of those motions changed the way that the molecule smelled to the flies. Now they thought it smelled disgusting and they avoided it. So isotopes change chemical behavior in all sorts of ways, including ways that a relatively complicated animal can sense. Let's now focus in on chemical isotope effects that have natural consequences that lead to sort of records that we can study in the environment. One that's a very straightforward one is that when I do a heavy isotope substitution in water, I make it heavy water. I replace the hydrogen atoms with deuteriums. The boiling point of that water goes up. That is, I have to make it hotter to evaporate it. And the reason is that the bonds between hydrogen atoms within the water are slowed in their relative motion, their vibration. Therefore, they're stronger. Therefore, they're more stable. It's harder to pull the water molecules apart so that you can evaporate. Because of this, I separate the isotopes of hydrogen during normal meteorological processes. So for example, when I charge the atmosphere with vapor by evaporation over the oceans, it's easier for me to evaporate the hydrogen-bearing version and harder to evaporate the deuterated version, the version with a heavy isotope of hydrogen. And therefore, moisture in the atmosphere is lower in deuterium content than it is the ocean. Then this difference gets accentuated through weather. When the moisture moves to some other part of the atmosphere where it used to be saturated, now it's supersaturated and it wants to rain, then it's easier for me to condense the deuterated version back out of the vapor phase because the water molecules stick to each other more strongly when they're deuterated. So I begin with less deuterium and then I preferentially pull it out of the cloud. And this acts as a kind of distillation, so that the deuterium content of the cloud and of the rain that comes out of it drops further and further the greater the distance you are from the moisture source. And this creates sort of regional geographical variations in the deuterium content of rain. Usually it's higher deuterium content near coastal regions where you're close to moisture sources, and lower in deuterium content in interiors of continents where you're further from moisture sources. This can serve as a kind of geographical forensic tool. Here's an example done by a scientist named Jim Elringer at the University of Utah, where a student who had just reached his laboratory from China agreed to have one of her long hairs pulled out of her head, cut into small pieces, and then analyzed for deuterium content. And starting with the freshest hair, okay, nearest to her scalp, It's poor in deuterium because Utah sits in the middle of the United States. And then the oldest hair in her head, near the tips, was higher in deuterium because she came from a coastal part of China before she moved to Utah. And so this gradient in the hydrogen isotope content in her hair is a kind of chronological record of her movement over the surface of the Earth, her geographical movement. And this is used for all sorts of purposes like that. The next tool that we're going to look at takes advantage of the fact that all molecules want to take in heavy isotopes to increase their chemical stability, but they compete with each other. Okay, they are not equally good at harvesting heavy isotopes. The most important competition of this sort in terms of applied tools involves a thermometer, a measurement of past temperatures involving the trading of oxygen 18 between the carbonate ion in aqueous solution and the water that it can exchange with. So if I start with the isotopically normal carbonate ion in the isotopically normal surrounding water, both of them are molecules that are undergoing internal vibrations, internal motions. And now let's imagine I have a heavy isotope of oxygen, an oxygen 18 atom, and I want to place it in one or the other of these molecules, and I watch and see what happens to the vibrational energy of the molecules. If I first put it in the water molecule, I slow the rate of vibration of the hydrogen-oxygen stretches, and I reduce the vibrational energy of the water, and the whole system now is more stable. Okay, I've lowered the total vibrational energy content of the system by reducing the vibrational energy of the water and leaving the carbonate the way it was. But if instead I make the other choice and I put the oxygen 18 into the carbonate ion, which is a stiffer bond with a larger number of ways of moving, I lower the energy of the carbonate ion more than I lower the energy of the water atom. And so the system prefers that you put the oxygen 18 in the carbonate ion. The system is trying to minimize its chemical energy. And so it wants the rare isotope to go wherever it can do the most good. It does the most good in the carbonate ion, so that is the one that wins the competition. Carbonate steals oxygen 18 from water. If this was the only effect that mattered, this would be a perfect separator. Carbonate would contain all the oxygen 18 in the ocean, and the water molecules themselves would contain none. But there's another effect that occurs in chemical energies, and that is entropy, or in the kind of entropy we care about, is randomness of isotopic distribution. If I were to fully separate the heavy isotopes into one molecule and the light isotopes into a different molecule, there's really only one geometrical way that that looks any different from each other, that I can make such an arrangement, okay? There's the pure version of phase A with all heavy isotopes and the pure version of phase B with no heavy isotopes. We call this a low entropy state. There's a small number of configurations that can have this composition. If instead I share the rare isotopes between the two phases, then there's many different configurations that can look like this. The rare isotope, say this rare isotope at this upper left corner of phase A, it might be where I've drawn it, or it might be next to it, right here in this yellow spot. They could trade places. Those would be two different configurations. And I'm sure you can imagine there's many different configurations I could draw. Lots of configurations means high entropy, high randomness. We know from basic thermodynamics that the free energy change of some chemical process, which is our measure of how advantageous it is, how preferred is that process, it reflects both the heat of the reaction, the enthalpy of the reaction. That's basically the vibrational frequency effect I was talking about before, minus temperature times the entropy of the reaction, the change in randomness. Because of this term being multiplied by temperature, it means entropy is not very important when things are cold, and it's very important when things are hot. And therefore, the difference in oxygen 18 content between the carbonate ion and water will be large at low temperatures. The enthalpy effect or the vibration effect wins, and it will be small at high temperature. The randomness or entropy wins. And it is this temperature dependence that lets us do geothermometry. It lets us measure the temperature of past climatic events by looking at the separation in rare isotope content between two materials that underwent one of these competitions, if you will. And here's an example of this being done in practice. So the vertical axis is age, showing you the history of the Cenozoic, and the horizontal axis is the oxygen 18 content in funny kinds of units. Don't worry too much about exactly what they mean, but they're a measure of how much oxygen 18 has been concentrated into benthic foraminifera, small shelly fossils that grow on the bottom of the ocean floor. And what you can, this has an inverted scale for obscure sorts of reasons. So basically, the oxygen 18 content of these shelly fossils early in the Cenozoic was relatively low, near this index value of 0 or 1. And then it rose to values of 3, 4, or 5 as the Cenozoic unfolded. And the reason for that is twofold. One is a cooling of the Earth. That is, the carbonate ions became more and more efficient at stealing oxygen 18 out of seawater. Second effect is that the ocean became distilled in its oxygen isotope content by removal of glacial ice to grow the ice caps. The trick in this field is, how do I balance those two? How do I tell the difference between temperature controlling the isotopic distribution and the ocean's oxygen isotope content? I would somehow have to go back in time and measure what the oceans were like to compare with the fossils at the same time if I were going to interpret this rigorously. And the way the community has responded to this uncertainty is to really only consider the older part of the data before the nucleation of the ice sheets as being a measurement of deep ocean temperature. And then the more recent parts of the record, there's some funny combination of temperature and ice sheet volume. And that's about as far as you can get. We'll return to that point in a moment. But before we return to whether we can do better in determining past temperatures, let's now move to another kind of isotope effect, biological isotope effect, sometimes called vital effects. That is the biological separation of isotopes. This is a very complicated field. Often it gets described in sort of simplified terms, but virtually all biological fractionation of isotopes involve multiple processes happening simultaneously. And we are really only able to observe and easily understand the net effect, the combination of all of them. One way to sort of organize your thoughts around this complicated subject is to distinguish between autotrophy, the fixing of carbon, say through photosynthesis, and heterotrophy, the redistribution of already reduced carbon. So we're obviously heterotrophs. We assimilate our food, something that's been previously fixed in, say, a plant or an animal that ate a plant, whereas plants are fixing their carbon through photosynthesis. Generally speaking, fixation, autotrophy, is very isotopically fractionating because you're changing the bonding environment of the carbon atom. Heterotrophy is, generally speaking, not very isotopically separating or fractionating because you're simply shuffling, redistributing already fixed carbon in biomolecules like amino acids and fatty acids and things like that. And the principle that applies there is the you are what you eat principle, that to first order, you have the isotopic composition of the food that you ate. So here's a sort of a short primer on the way the photosynthetic fractionation works in the most common land plants. CO2 in air has about 1% carbon-13 in it. And when it's fixed by the so-called C3 plants, which are basically broad leafy plants, there are two processes that take place in the fixation. One is the diffusion of the CO2 through pores into the interior of the leaf. That's a process we talked about earlier. And then a chemical fractionation that I haven't gone into, but is more complex, where the CO2 is bound to the RUBP molecule to make sugars. And that's a second fractionating process, a chemical fractionation. And it's the addition of those two effects that leads to about a 2.5 or 3% depletion in the carbon-13 content of plants compared to air. There's another form of photosynthesis, it's more common in grasses, where CO2 diffuses into the interior of a plant. It's partially fractionated in its isotopic content that way. The C12 enters, the C13 is left behind. But then, before it can be photosynthesized and turned into sugar, it's grabbed and turned into an aqueous complex and pumped to the place where the actual chemical reaction will occur. And where the photosynthetic fixation occurs, it's basically complete. And if you do a process to completion, like if I evaporate a glass of water completely, I can't separate the isotopes. I took all the isotopes that were in the glass and I put them all into the air. Same here. I take all the isotopes that I pump to the place where the Rubisco reaction, this fixation reaction happens, and I use them all. Because of this, I see only the diffusive fractionation. It's less fractionated. It's more similar to the air. Okay, and then there's other biological processes like fermentation might take pre-existing biomass and then microbially change it. We're not really going to get into that today. So how do we use these sorts of signatures as tools? Here's an example from ecology where we're studying the diets of migratory elephants. So anybody who's lived in a region that has elephants in them knows that they like to move around and they'll eat a farmer's field if you let them, or they'll eat leafy plants in some seasons, grassy plants in other seasons, whatever's available. They move around a lot and their diet varies a lot over the year. So what's shown here in these dots is the carbon isotope content of the hair collected from an elephant, sort of like this graduate student's hair, except now we're looking at carbon instead of deuterium. And then a function that's related to studies of the dietary assimilation in elephants shows what the inferred diet was like. That's this heavy line. And then the fraction of C4 plants, grasses, versus C3 plants, like leafy bushes and shrubs and things like that, is shown in the gray at the bottom. And basically, when the elephant is eating grasses, it's eating lots of these C4 plants that have that separate the diffusion from the photosynthetic fixation, and the carbon isotope content is closer to the atmosphere. During the wetter seasons, before and after the winter dry season, you're seeing lower 13C contents, reflecting the eating of plants that combine the diffusive and the chemical fractionation step, and they both get expressed. Here's another example where this is used not for ecology, but for criminal forensics. So there's a cross plot that shows the carbon-13 concentration in cocaine that was captured by law enforcement agents who were working in South America. And then another kind of fractionation, the nitrogen isotope separation, that also occurs during plant growth. And what they've done here is, instead of trying to interpret the small variations mechanistically in terms of exactly how the photosynthesis is working, it's just forensic fingerprinting. They just want to document how different regions of the sources of these drugs differ in their carbon and nitrogen isotope contents from others. And then they can take a sample that was recovered from a drug dealer or whatever and map it back onto the place that it came from, rolling up drug trafficking organizations. Here's another example of using the biological fractionation of isotopes as a way of sort of forensically recognizing materials working through a food web. So here's an experiment where a person was fed on corn, on a mixture of like corn and soybeans and things like that, with some intermediate carbon-13 content that reflects the average of their diet. And then at the beginning of an experiment, they forced this person to drop all the leafy vegetables from their diet and just eat grassy vegetables, okay, like corn and things like this. I don't recommend it. It's not healthy for you, but it's what they did in this experiment. And then after doing this, the person's hormones in their bloodstream drifted towards this new food source. Okay, and that shows that the hormones in your blood are just following the "you are what you eat" rule. How do you use this? Here's an example. This is a guy named Floyd Landis who won the Tour de France, which just finished a week and a half ago or something like that. So I thought it was a nice example. So a number of years ago, Floyd Landis won the Tour de France, and they sampled his blood afterwards and saw a hugely elevated testosterone content. And he said, well, it's not my fault. Just how I am. I have lots of testosterone in me. And they couldn't really argue against him. The concentration of steroids in your bloodstream are so variable from individual to individual that you could never convict him of cheating just by measuring his testosterone content. So what they did next is they measured the carbon-13 content of all of his other hormones that don't really have anything to do with athletic performance and compared it to the testosterone in his blood, which was lower in 13C content, more similar to the C3 plants. This is because synthetic testosterone is made from phytosterol precursors that come from soy. And so there was a difference between his average diet and the testosterone he had injected, and that's why he had the Tour de France title taken away from him. So there are a few more things that I wanted to talk about, more complicated, newer sorts of tools, but we don't have time. So I'd rather take questions from you. I was going to tell you why dinosaurs have, whether they're warm or cold-blooded, but you'll just have to learn that by reading the funny papers or talking to me afterwards. So I'm going to leave this last slide up as sort of a reminder of what we talked about, and I'll stop sharing and take any questions you might have. Victoria.
Yes. Good afternoon. Thank you very much, John, for the insightful lecture. So my question is based on the slide you showed images of benthic foraminifera and how you can use the oxygen isotope to interpret or infer temperature. So I want to find out, how do you detect how do you use carbon isotope to, how does carbon isotope reflect climate change?
Yeah, so the carbon isotope contents of the foraminifera, both the benthic ones that grow on the ocean floor in the shallow part of the sediment, and the planktonic ones that float higher in the water column that have photosynthetic symbionts in them. People study the carbon isotope difference between those two types of organisms to study the strength of the biological carbon pump. So the biological carbon pump basically involves photosynthetic organisms in the surface ocean fixing carbon. When they fix carbon, that creates a deficit of dissolved inorganic carbon in that water, and it forces ingassing of atmospheric CO2. If the planktonic organisms die and then remineralize, get respired, digested by other organisms, shallow in the water column, there's no net effect. Okay, the carbon, you don't really get to remove carbon from the surface ocean that way. You're just cycling it quickly. But if the planktonic organisms sink into the deep ocean before they are remineralized, before they're respired, then you're sucking carbon down into the deep ocean where it can be stored for thousands of years. And so the contrast in carbon isotope content between the surface ocean and the deep ocean reflects how strong this biological pump is. And people map this all around the ocean basins and through time to try and understand how ocean biological activity and then circulation traps carbon in the ocean during cold climate times and releases carbon back out of the ocean during warm climate times.
Thank you.
Yeah, good question. Okay, anyone else? Well, thank you all for your attention. If you have any questions later on, something occurs to you, or you'd like to know more about the program at Caltech, please feel free to write to me.
Great. Thank you so much. That was great.